Okay, this is the discussion of the cell cycle. Cell cycle is basically the life cycle of the cell. Consists of these two main parts. First one's interphase, and the other one is M phase, and M phase is made up of mitosis and cytokinesis. So let's look at those parts. Interphase is when the cell is just doing its job. The cell's growing, getting to be a big boy or girl cell, doing whatever function it is. So if it's a stomach cell, it's doing what stomach cells do. If it's a blood cell, it's doing what blood cells do. Okay. During interphase, there are three phases, three subphases. G1 and G2 are called gap phases. And during these, the cell is still doing its job because it's interphase, but things like organelles are replicated. And during S phase is when the DNA is replicated. Okay. Mitosis creates two new daughter cells, and the two new daughter cells are exactly like the original mother cell. Mitosis is division of the nucleus, so that's when we divide up the chromosomes into two new nuclei. And then cytokinesis is division of the cytoplasm along with the rest of the organelles. Phases of mitosis: prophase, metaphase, anaphase, telophase. And these are just very brief snippets of what really happens in here. So you can take a look at these. There are no specific questions on the test about what happens, but you should know that these are the four phases. And for the lab, you're going to have to know the four phases. So the big one is that metaphase. The chromosomes are lined up across the middle of the cell. And I'll show you a picture of that. Anaphase is when they start to pull away from each other. And telophase, and two new nuclear envelopes forms. Okay, and this gives you those major events in there, so you can look in this and write it down in your notes. Here's mitosis. What happens, we're going to start here with this brand new cell. And this brand new cell is going to be in interphase, where it's just going to do its job, do whatever it's supposed to do. But then in S phase, the DNA is going to replicate. So you're going to have copies of your DNA. And don't forget about these centrioles that we learned about. And the centrioles only show up during animal cell mitosis. Okay, during prophase, you can see the nuclear envelope starting to break down there. So here the nuclear envelope is breaking down. Don't worry about late prophase. Metaphase, the chromosomes and their copies are lining up across the middle of the cell. Anaphase, they start to pull apart. Okay. Telophase is when the new nuclear envelopes are reforming. And then the cytokinesis is when each cell is going to get a share of the organelles in the cytoplasm. Okay. Cytokinesis, it does begin during anaphase and continues all the way through telophase. The new cells will have the same DNA, but could be different in size based on how many organelles each got. Cell division is controlled through numerous pathways. But these are just some of those general pathways. We're not going to talk about specific ones. First of all, at the end of the chromosomes, there are things called telomeres. And those telomeres fall off every time the cell divides. And what that creates is, when the telomeres are gone, the cell can't replicate the DNA anymore. And that prevents old DNA from showing up in cells. Okay, and then there's all these things called cyclins and cycle-independent kinases, which are proteins that control the cell cycle. Cells generally divide because their surface area can't support the volume. And this happens when cells get to be too big. So when a cell gets too big, and when cells are small, I should say, the surface area and the volume are in a ratio that allows stuff to enter and leave the cell to support the cell's metabolism. When a cell gets too big, the surface area can no longer support the volume because volume grows faster than surface area. There are hormones and other growth factors that are external controls for cell division. So for example, growth hormone. Growth hormone can tell cells, hey, you need to divide because we need more muscle cells or bone cells. Contact inhibition. Normal healthy cells have controls on them, so when they touch other cells and get crowded, they stop dividing. This way we don't have cells growing out of control. And then any loss of cell cycle control can lead to tumors, whether they're benign or malignant. Okay, two types of tumors. Benign, they stay in a local area, they don't spread. The malignant ones metastasize, which means they go to other tissue and it can invade that tissue. Two major types of genes that cause cancer, oncogenes, are when there's a damage to genes that control the cell cycle. Okay, and then so that can cause it. And then tumor suppressor genes, if those are damaged, so they're inactivated through DNA mutation or they're taken out completely, then they can't regulate mitosis. Cancer cells are immortal. I highly suggest reading the book, The Immortal Life of Henrietta Lacks, because it's just a great discussion about this, and I think you'd really enjoy it. So cancer cells don't stop dividing. They divide forever. Cell differentiation. Cell differentiation is what allows a stem cell to become many different kinds of cells. So stem cells can divide to form two new stem cells. Okay, and that's just stem cell self-renewal, where one stem cell divides into two stem cells. They can also divide to form a stem cell and a progenitor cell. And we see this with our bone marrow. We see this with our gamete production to make our sperm and eggs, and can differentiate as any of many cell types. So stem cells can become a lot of different types of cells. Progenitor cells, though, are limited into what they can become. So they're partially specialized stem cells. Progenitor cells come from stem cells, but they're really restricted in what they can become. Progenitor cells are quite often called these committed cells. Totipotent daughter cells that can specialize to become any cell type. For example, fertilized egg. So that zygote is a totipotent cell because it's going to become many thousands of different types of cells with different functions. Pluripotent means it's limited into the number of cells it can become. So the totipotent cell comes first, and the pluripotent cells come from that. So this is an example of a stem cell dividing into another stem cell, and the other daughter cell is a progenitor cell, the myeloid progenitor cell, which becomes some of the white blood cells. Okay, we've already talked about this. So just refer to this diagram when you're looking over those notes. Cell death. Programmed cell death is called apoptosis, and it's when cells are programmed by the genes to die. And it could be just part of normal development. So for example, when the fetus is developing and those webbed fingers and webbed toes need to go away, there's programmed cell death in there to get rid of that, which is mentioned right here. And then there's also programmed cell death when we have damage to our skin and from a sunburn, and it causes those cells to die off and be gotten rid of, and then new cells can form underneath. Okay, cell necrosis is from damage, and it's not a normal process. So this is, you cut open your cells with a knife, you cut your finger. That's not programmed, that's just you killed the cell.